I'm from Max Planck Florida Institute. I'm going to talk about this postsynaptic biochemical computation. But just tell you a little bit about Max Planck Florida Institute. This is just only one Max Planck Institute outside Europe, and we have a group of people working on neural circuits and like development, plasticity, and function of the cortical and other part of the brain circuits. We have about 7 groups, 2 directors, including myself, and 5 independent research group leaders. Kind of very kind of unique aspect is kind of funded by both Max Planck, Florida State, and also some funding from NIH through grants and kind of thing. So it's really close to here, actually, right? It takes only 3-4 hours flight from here to Florida. So if you have chance, it's really nice institute to come. Anyway, so we have been working on, my group is working on intracellular signaling in neurons because we think that it's very important for the brain action in long time scale. So as you probably know, our brain acts on different time scales, right? So and a lot of function transmitted by electric or synaptic activity is in millisecond time scale. But a lot of other aspects, right, learning memory or our behavior adaptation, these basically happen in much longer time scale from seconds to days. And we think that, well, a lot of evidence suggests that these are coming from intracellular signaling in each neurons. So if you look at each neuron, so in this case it's a hippocampal neuron, the cell is huge, right? The neuron is really huge cell with a very complicated morphology. And if you zoom into, for example, receiving side of the neuron called dendrites, right? They have extremely complicated branching pattern, right? And these small structures called dendritic spines, they receive information from other part of the brain. So they at other neurons. So, and we have about 1,000 to 10,000 synaptic inputs per neuron. And these basically synaptic input, of course, have important function. But we are interested in is actually that this role of dendritic spines for much longer time scale signaling. For example, spine is known to change its structure, right, during learning and memory. For example, formation of spine, enlargement of spines occurs when you memorize something, and these are stabilized to maintain the memory, right? So this kind of structure plasticity is considered to be important for our capability to memorize things. And consistent with the important function of dendritic spines, there are a lot of studies suggest that a lot of mental diseases, it's just indicated here, are associated with abnormal spine morphology. So it is kind of important to learn how spine structure and the structure plasticity is regulated. We have been working on one particular type of structure plasticity extensively. We also work on other plasticity paradigm, but this one is kind of interesting. So you can see that activity of this spine in one spine causes enlargement of the stimulated spine. And you can see that this occurs only in the spine that is stimulated. Nothing happens in other spines. It's kind of spine-specific synaptic plasticity. And this volume change occurs right after stimulation, right? So in this case, we use glutamate uncaging, but we observe exactly the same enlargement with electric stimulation. Basically, we get enlargement lasting more than 40 minutes. So there is a sustained phase which seems to be important for functional plasticity, like glutamate sensitivity increases in the spines over more than 30 minutes. It actually lasts more than several hours. And only in stimulate spine, we observe increased glutamate sensitivity in this. So basically, this spine shows increased glutamate sensitivity. Surrounding spines don't show this kind of thing. So this is a long-term potentiation that just introduced by previous speaker, but basically is associated with structural change of the spine and number of receptor change. And we think that these are essential part of brain plasticity. So again, our question is how are the structure and function of the spine regulated through intracellular signaling? Now this becomes a big problem because actually each neuron expresses like tens of thousands of proteins, and they are connecting to each other, and all proteins are important actually. So this is actually a huge network existing in each synapse. And we need to basically understand what's going on in this very tiny volume of spine. Actually, each spine contains only 0.1 femtoliter of volume. So it's actually extremely difficult biochemical problem we have to solve. So I show you today two topics. So monitoring activity of proteins within this tiny volume of the spine and manipulate them to see how activity of signaling proteins regulates structure and function of dendritic spines. Okay. So for monitoring side, we are using optical technique, and we use a specialized microscope called two-photon fluorescence lifetime imaging microscopy, which we developed a long time ago, but it happens to be extremely useful tool for us. So briefly, so this is a combination of two-photon fluorescence microscope, which is kind of standard technique for imaging deep tissue, like brain slices. We often use brain slices or intact brain. Both of them requires usually two-photon microscope to image. But fluorescence lifetime imaging gives us kind of information about the fluorescence coming from the fluorophore. And basically, basically when we excite fluorophore short laser pulse, we can measure the decay of fluorescence occurs in nanosecond time scale, and which happens to be sensitive to the interactions between different fluorophores. So using this fact, we can we can produce sensors that can basically transfer, well, actually that can translate activity of protein, like kinase activity or small GTPase protein activity occurs in tiny volume of spines, right? And we can read out optically the activity of proteins. Okay, so we made a lot of different sensors, like CAMK, a lot of kinases, yeah, ARC, PK. I don't know if you know these names, but these are all molecules important for synaptic plasticity. Now, if you can image the activity, now we can actually map where they are activated, what's the time scale they are activated. So here I just draw the signal transaction in different time scale we measured, right? From 0.1 second, which is relatively short, right? Electric activity is much shorter, but actually 0.1 second is basically time scale of calcium. Calcium increases and decays almost immediately within 0.1 second, which is transmitted by different signaling molecules over 10 seconds, which is further relayed by these molecules over 10 minutes. You can see probably actin here. Actin is a cytoskeletal protein, which basically changes the basic building block of dendritic spines. Right? So I show you several proteins today so that you see how it looks, how our measurement look like. So for example, we have been measuring activity of a kinase called CAMK2, calcium carmogene dependent kinase 2. Okay, just remember this name, CAMK2. Right, this is a kinase activated by binding of calcium carmogene. Okay, so it's a calcium sensitive kinase. To measure this activity, we are using a sensor basically made by CAMK2 itself, attached to two fluorophores, right? That are reporting us the distance between these fluorophores, right? So we measure again fluorescence lifetime, excite fluorophore here and measure the decay of the fluorescence. And you can see actually open form decays slower compared to the closed form, right? So we can measure the activity of the protein using fluorescence lifetime. If we actually scan while we are measuring, this is supposed to be a movie actually. Okay, good. then we can generate generate activity map of the protein. So this is a single spine. Again, it's a 0.1 femtoliter, right? And we have synapses 1, 2, 3, 4, 5, 6, 7, 8, 7 synapses here. Only one synapse is stimulated. You can see actually that protein is activated at the moment. Actually, a little bit before the spine structure plasticity occurs, it's activated. So this is a time course of activation. We measured with the lifetime. It's activated, and immediately after we stop, the stimulation decays. Okay. And if you align calcium and CAMK2, okay, so maybe you remember calcium activates CAMK2. So calcium decays again very fast within 100 milliseconds. Now CAMK2 basically integrates calcium activity inside the spine. So CAMK2 acts as leaky integrator to basically integrate the calcium signal over about 5 to 10 seconds. Right. So this is kind of important role of CAMK2. Calcium is so short, it doesn't act as memory. But CAMK2 is important for memory formation. Basically, it's integrator over 10 seconds and transmits signal over to more longer time scale. I'll show you one more example, which is actually different, little bit different system, BDNF, TrkB. These are ligand and receptor in the neuron. And basically, when this receptor binds to this ligand BDNF, they are dimerized, and this actually is a kinase. There's a kinase domain here, which kinase basically phosphorylate each other and make phosphorylation like this, which recruit proteins like SHIC, PLC, and other proteins. You don't need to remember these proteins. But this causes spine structure change, learning memory, and it's important for a lot of things. Okay, but nobody knew actually where it happens. So we made sensor. Our sensor is made of TrkB itself, this receptor itself, fused to GFP, right? And this binding protein fused to RFP. Okay, so basically they come close to each other when this protein is activated. So we can measure actually using fluorescence lifetime. And this is a measurement, right? You can see that it's activated, certainly. So this is before. So we are stimulating the spine. You can see that the color becomes red. It means it's activated. Or the color code is wrongly, actually. Red is active. And basically stays active for a long time. So you can see actually that there are different time scale from previous kinase, right, which is CAMK2. So previously I talked about this one, but now we are talking about this one. It's activated within about 30 seconds, and activation lasts for a long time. Okay, so basically this tells us that this kinase actually is activated in much longer time scale. Actually, BDNF interestingly is released from the spine and from the stimulated spine and binds to the TrkB receptor on the same spine. And activation lasts more than 30 minutes. Okay, so repeating this kind of measurement, we can basically map also on this diagram how activation spreads from calcium. So calcium here, we measured about 20 different proteins in different time scale, and we can align them what's the order of event occurs in dendritic spines. This is very simplified. We have only one spine nucleus, but you can see actually, so calcium starts signaling, causes release of BDNF, which activates these proteins. These are proteins important for changing the cytoskeleton. And nucleus changes, of course, gene transcription program. And these are internal membrane signaling, which is also important for membrane trafficking and receptor traffickings. Okay. Now, so you see that this is very powerful technique to image activity of proteins. But to correlate this activity and connect to each other, first of all, and also connect this event with the actual learning and memory, we need to be able to manipulate these proteins quickly. We have been working on different type of inhibitor which can be activated by light. Okay. So usually inhibitor, of course, it's very difficult to apply in actual live animals, right? But it takes a long time, sometimes days. But these proteins act on much faster time scale. So we decide to make an inhibitor that acts almost immediately. So So this is basically collaboration and with my former postdoc Hideji Murakoshi. So we basically developed light-inducible kinase inhibitor CAMK2. Maybe you remember this protein. This protein is activated in response to calcium in the spine. Okay, and we attached kinase inhibitor, peptide inhibitor, with this LAV2 domain of phototropin, which is a protein derived from plant, and basically changes its conformation when it absorbs light. Okay, so when it absorbs light, it opens up, it inhibits CAMK2, and it spontaneously comes back in 40 seconds. So this actually works very well. So for example, this is in slices. We again are estimating one spine here. We get structure plasticity like this. In the neuron expressing this inhibitor. So, but when we apply blue light just for 1 minute, right, then we can stop the spine to grow. Okay, the spine growth can be completely inhibited, and this timing dependent. We can because CAM-kinase is active only for 1 minute during the stimulation. Basically, if we shift the application of this blue light, we can completely rescue the synaptic plasticity. So now we have a tool to manipulate the protein in the neurons, right? Manipulate the activity of protein kinase in the neuron, and this kinase is important for recording memory. So we tested if we can stop the recording, memory recording, in actual animals. So with Myun Shin in my lab, she's a postdoc, we decided to work on this. So basically, we developed a behavioral paradigm called, actually it's already developed, we adapted the behavioral paradigm called inhibitory avoidance. It's kind of one of the tasks to test fear memory. Right? Basically, mouse is located in a chamber separated in 2 rooms, one bright and the other dark. And at one moment, the room separating this opens. Now, usually mouse prefer to get into the dark room, so they get in. But once get in, the door closes and mouse gets shocked. Okay, so mouse doesn't like it. So if you repeat this, mouse really learns very well, so they never come into the dark room. And the brain region required for this is several, but this one brain region important for this is called Amygdala, which is a center for a lot of emotion, including fear. So we infected this inhibitor, light-inducible inhibitor, and these neurons, just pyramidal neurons, excitatory neurons, and put light guide, right? It's a little bit destructive. We made a huge hole here, but anyway, we put light guide, right? And let them behave. Actually, they can behave very well. Okay, you believe or not. Okay, so this is the performance of the mouse. We basically measure how long it takes for them to get into. And control is just we use control virus. Okay, so we have similar kind of hole and similar amount of light. So almost all animals, like among 20 animals, almost all animals did not get into the room right after 1 hour. Okay, sorry, I forgot to put when we apply blue light. We apply blue light from the training, right? And finish for 1 hour and finish blue light and then do memory test. So this one, with the control virus, almost all animals learned not enter the room. Okay, at 600 seconds we give up. Now, for actual active compound, you can see that almost all animals did get into the room. So they actually don't, well, we interpret that they actually don't remember the shock. Or actually they don't feel the fear, at least, over this, get into the room. So they really don't have any fear into the room. They rush into the room. Now, interestingly, this protein is activated only during the synaptic stimulation, right? So if we start the blue light after the training, we do 2 shock here, right? And right after that, we start the blue light application. and then do memory test. Then actually again all animals learn. So we can basically test when this kinase is activated during the learning paradigm and when actually memory is encoded. So it seems like in this case CAMK2 is activated during the training and they play some role in encoding memory. It's too late to inhibit this kinase after the memory is established. Okay. We have time. So we also team up with Wenbia Gan, a professor in NYU, and his postdoc, Avital Adler, together with Myung Shin, to test actually if this inhibitor can be used for different paradigm, like motor skill learning. So Avital developed a kind of very interesting motor paradigm, motor skill learning. Right? Basically here, mouse head is fixed here so that we can image the neurons in neuron activity in the brain, right? In the motor cortex. And rear limb is on this bar, but front limb needs to be on this treadmill. And actually treadmill sometimes moves. And so you can imagine this is kind of difficult task. You really can't walk on this moving treadmill in the beginning. But within 20 minutes, actually the mouse start to learn how to walk on this kind of weird environment, and they can do very well. So this is a footstep. So initially they are very bad, right? Just kind of sweeping, they have no idea what to do. But then after 20 minutes, they can just walk very nicely. Now this is associated with the change in the motor cortical neural circuit. This is activation of neurons imaged with calcium. So you can see actually that. So this is a motor. The treadmill running for 40 seconds and neuron activated at different timing before the training. After the training, most of the time neurons actually, a lot of neurons start to be activated at the beginning of the motor, beginning of the treadmill movement. So we think that this kind of shift is a motor circuit change, but we wanted to test actually this plasticity in the motor circuit, right, the brain circuit, and plasticity of the behavior, in this case, motor skill learning, can be stopped by CAMK2 inhibition in this particular neuron. That seems to be important for this behavior. So we go for pyramidal neuron. It's a specific neuron in the motor cortex and express it. Okay. So first of all, okay, 1 minute. So first of all, this is a neural circuit plasticity with the light. And with this control virus, you can see that it actually motor circuit plasticity completely stopped. Behavioral performance, it's percent structure running, right? So in control virus, we get big improvement, while when we just inhibit this kinase only during the training, we get complete inhibition. So this way, basically, we can find which neuron is undergoing plasticity and which kinase doing when during the learning and memory. So this way, we hope that we can connect actually the behavior, synaptic plasticity, molecular activity, synaptic plasticity, and circuit plasticity, and behavior, learning, memory. So with that, I finish my talk. So this, of course, done by a lot of people, and I thank all people who actually did all these experiments. And because I run out of time, I just thank you for listening.